All students, am I audible?
Yes, sir, you're audible.
Okay, so welcome back. So I think you are joining. So we can start after 5 minutes. Okay, so now number is significant. We can start. Am I audible? Am I audible?
Yes, sir, you're
Devanshi,
audible.
Yes, sir. Yes, sir.
Devanshi, Devanshi.
Yes, sir.
Am I audible, Devanshi?
Yes, sir, you are audible. Sir, am I audible to you?
Okay. So now we can continue. Okay, so we have done most of the enzyme, and now we are to our last topic of this module. And your speaker are not working. Okay, so my speaker is not working today. Might be, I don't know why, what issue is. So if any question that you want to ask, so please type in the chat box. Okay, so thing is that I was referring that whatever we discussed about the molecule or our enzyme. So now the issue is like, as you know, when we were talking about ligases, at that time we mentioned that and also the restriction endonucleases, endonuclease produce two types of the cut. One is a sticky cut and second is the blunt cut. So, and while you are performing your cloning experiment, depending on what restriction site is present in your gene, you are going to get that type of fragment. So, if your gene or your DNA do not have any of the restriction site which is going to produce a sticky end, so then what is going to happen? Then in that case, it is going to produce, it is going to give you a blunt-ended molecule. So in blunt-ended molecule, what we should do? We have to ligate. Either we should proceed for blunt-ended ligation, but the issue with the blunt-ended ligation is that it is a bit slow as compared to the sticky end one, and also direction you cannot You can't make your joining in a proper direction because both the sides, both the ends are going to behave in the same way. So we can't say that the direction in which we want the cloning should occur is happening in that direction or not. So this the directional cloning is an issue when you are going to work for the purpose of expression. So the two different concepts we are going to deal in the future classes. That one is the simple cloning, and second is the expression. So the cloning means producing a similar type of a DNA fragment or similar molecule, but the expression means you are going to get some product. Like your gene is going to code for some protein. Means you want after the cloning transcription should occur, and after the transcription your translation should be there. But if the direction is reversed, so then your entire gene is going to be reversed. So the protein that you want is not going to be the same. Okay, so as I was referring you, the compatible sticky ends are desirable on DNA molecule to be ligated together in gene cloning experiments. So that's why we need a sticky end. But as I mentioned, it will not happen always because of limitation of your restriction nuclease. So for to resolve this issue, like we are having a blunt end, then can we make this blunt end to a sticky end? So for that, three different strategies being used by which we can make a blunt end to a sticky end. So the first strategy is a linker. What we can do, you can add a linker. Linker as a name is there. Linker is going to be, you are going to link something which is going to assist you in the linking. Simply that is for the ligation. So what the linker is? So linker is a short piece of double-stranded DNA. So here you must remember when we are going to distinguish two different things. As I mentioned, we are going to talk about linkers and adapters. So linkers, what are linker and adapters both we are going to use for generating a sticky end. But here, what way these two are going to differ, that we are going to discuss. First, this is a linker, means it is a double-stranded DNA. Okay, one thing. And when we are saying double-stranded, it means it is again going to be a blunt end. So it is not going to be a sticky one. Okay, so this is the feature of it. And one second feature is going to be nucleotide sequence. You are knowing this nucleotide sequence, and knowing this nucleotide sequence means it is going to we have some restriction site that you desire. Like in a case where we can happen that two DNA molecule that we want to join, one DNA molecule is going to be have a sticky ends, like your vector that we are talking about. If vector you produce a cut with a BAMH1 or EcoR1, they're going to produce sticky ends, but the fragment that you want to insert there, that is going to be a blunt end. So in that case, you want to make your segment that you want to insert to be a sticky. So what you can do, take a double-stranded DNA molecule of a non-nucleotide sequence that you can synthesize into test tubes, and that will serve as a linker. So this is a, you can see it is a double-stranded one, and sequence is known. What is going to have? It is going to have a site that is a BMH1. So if if you restrict digest this sequence, it is going to produce a sticky end. So this linker you are going to link to your segment. So now you will say that, sir, this is also blunt end, and the segment that we are having, that's also blunt end. Then how this linker is going to help you? As earlier you mentioned that sticky ligation of blunt ends are a bit slow. So why we are going to incorporate one more step where again the ligation is going to be slow. So thing is that in this case is that the amount is not a limiting factor. Amount of your fragment of interest or gene of interest is a limiting factor. You are going to have a definite amount of it. But here in the case of linker, amount is not definite. Amount is indefinite. As you mentioned, you can synthesize in test tube. So whatever amount you want, you can synthesize it. And it is a small fragment. As you can see here, it is just not more than 12 or 15 nucleotide long. So it is very short. So synthesizing this small fragment is a bit easy so that you can do. So how it is work, how you're going to use it. It is a blunt end one, as I mentioned that this is a feature of it. and it contains restriction site. Any enzyme of interest that you need, you can use that one. For example, the BAMH1. DNA ligase attaches linkers to the end of the blunt-ended DNA. So same thing that you have to do, you have to ligate it utilizing the DNA ligases. Blunt-end linker can be made in large amount, as I mentioned you. When the large amount is there, when you are adding large amount of it, in end you are going to result with the efficient ligation. But in case of our normal vector and insert, amount is a limiting factor, so you can't perform your experiment with the large amount. So this is the thing how we are going to do it. So blunt end in one fragment you are having. How many fragments are there to ligate it? So you can see a large number are there, so they can ligate it. So you may ask us in what direction is going to be there. So for by direction, any direction is going to be linked. We all know that these restriction sequence uh, are palindromic in nature. So whether it will read from 5 prime to 3 prime or a reverse direction, both will need to play one role. If you say that if to this fragment more than one linker are going to be joined, that's also not going to harm you, because when you are digesting it with a restriction enzyme, the restriction enzyme is going to remove all excess amount of linkers which are joining to it. Okay, so now this is the use of linker. As we already mentioned, the main use of linker is making sticky end. So more than one linker is attaching here. So when you digest it, it is going to clip off. So finally, what you're going to get? You're going to get only one sticky end. Okay, not more than one. Okay, so these are going to be cleaved off. These are the cleaved linkers. Okay, so ordinary DNA fragment carry BAMH1 sticky site. So this is the use of your BAMH1 or your sticky linkers. Okay, so the modified fragment is ready for the ligation, so you can use it for the ligation to site that is of BAMH1. So as your vector is going to have a BAMH1 restriction site. So this is a linker. Okay, but a problem is there in linker is that the problem is uh, potential presence of restriction site within your gene. So if your restriction site is present within your gene, so that linker you can't use. So you need to design your linker a proper way. So you should consider what would going to happen when you are going to introduce one or more antigen production site. Okay, so that you should think of. So this is the limitation of it. So here you can see section step need to be cleared off, produce sticky ends. So like if like this, as I mentioned that uh, many sticky ends are going to be incorporated. So if your restriction enzyme doesn't digest it completely, so in that case it may sustain. So that is the one thing. And restriction step you needed it again. So one more step you are incorporating. So that's also a drawback of it. The resulting fragment will have correct sticky ends, but that is no consolation gene to be contained in blunt end fragment has now been broken into pieces. So this is the one thing that I mentioned that as we are saying that is being linked, but we can't assure that every of your fragment is being removed. Okay, so next type of thing is the adapters. So now how these adapters are going to differ from the linkers. So linker, as we mentioned, they are going to be double stranded. So here adapters are, as the name indicates, they have to adapt. So adapt means adapt to what? Adapt to the sticky conditions. You are changing your normal, what we say, uh, your blunt to the sticky one. So what their type of thing they are going to be? They are going to be by themselves sticky. So second method is the stick blunt ended molecule is designed to avoid this problem. So the problem that we are saying that the second time the second restriction digestion that we are performing, we don't need that. So for that, what we are doing, we are synthesizing short synthetic oligonucleotide. It is already having one sticky. So already you are having a sticky. It's not like linkers one, which was a blunt-ended one, and you have to restrict digest after linking. So what you are doing here, simply you can say that the linker that you are having earlier, that linker you are restrict digesting and after the restriction digestion of the linker, you are going to making that adapter that you are joining. So say that is called adapter. So here you can see this is an example of adapter. So it is having one blunt end, second is going to be sticky end. So from this blunt end, you are going to join with your DNA. So then it is going to lead to incorporation of a sticky end. So the idea, of course, to ligate blunt end of adapter to the blunt end of the DNA fragment. So here the blunt end ligation, it's not from the sticky end. So if the sticky blunt end are ligating, so sticky ends are available or sticky ends are overhand. So these overhand sticky ends you can utilize for the ligation. So ligation is from the ligation of adapter to DNA is from the blunt end, but ligation of adapter DNA fragment complex is going to be from the sticky ends. Okay, so this is the new molecule going to be the sticky end. So now things how we are going to be better and what are going to be a drawback of it. So drawback is as we are talking about in case of your linkers were that there is going to be from double strand one. So here you can see your adapters may have a complementary sequence, and these complementary sequence may lead to the formation of dimers also. So that's going to kill the job that we want to get from the your adapters. Like you need to produce a sticky ends, but if the dimerization is going to be occur, so the adapters that we are talking about, if they are self-complementary, as we all know that this is the issue with the your uh, the sticky end. So in that case, the job that we want to that adapter is going to serve you is going to be ended. So we will now, if such things are going to happen, in that case you again need restriction digestion to produce sticky ends. Okay, so the purpose is being defeated as for using the adapters. So in that case, we are using linkers to do our job. Or if both approaches are not going to work, then in that case we can use a third approach, and that third approach is the utilization of terminal transferases. So as we have discussed in previous class, what are the terminal transferases? They are the enzymes which are going to transfer a nucleotide at the 3' end of to become a sticky. So in such cases, we are not going to incorporate a large, long sticky end. So here just we are incorporating one or two nucleotides. So just like when you are restriction, if only U overhang is there, and if just one nucleotide U is there, and in your gene T is there, so this TU pairing is going to be occur, and this TU pairing is going to facilitate or further enhance the ligation. So this is the thing that we can use and to generate the sticky ends. So now we have uh, okay, students, please speak so that I will be able to know that my speakers are working or not. Uh, Devanshi,
Yes, sir.
uh, Devanshi.
Yes, sir.
Okay, I'm restarting the meeting, so please remain in the meeting. So I'm just quitting the MS Team so that might be it's MS Team issue.
Okay, sir.
Okay, so now this forum is open for the discussion. Devanshi, please speak. So now to verify that my speakers are working. Devanshi,
Sir, at my end, your voice is still breaking. I don't know. It is
but
my
but
issue or
but now at my end your voice is bit much very clear. Earlier I was not getting your voice. Then
okay.
it may be at your end. So now I have done with the module one, and so first we should I will take your question through from today's lecture that we talked about linkers and adapters or generating sticky ends. So if you feel that any difficulty or anything that you didn't understand from these adapters and linkers, so
Excuse
please
me,
ask.
sir.
Yes.
Sir, can you repeat the adapter part that you have explained?
Okay, so what you didn't get from the adapter, so first let me know, so then I will tell you
Sir,
again.
the second point in your PPT.
Okay, so you are talking about adapters. So which point you are referring?
Yes, sir, second and the third point.
Okay, so short synthetic oligonucleotides, this one.
Yes, sir, and the third point.
Okay, so this is the as I mentioned you in the previous slides when you're talking about the linkers. So linker we mentioned that they are again short synthetic nucleotides, but they are double stranded. So you can see the previous one. Sorry. So you can see here in this case of linker. So linkers are also synthetic one, but they are double stranded. So there is no sticky end in them. Okay. But what the linker are going to be? What are the adapters are going to be? Adapters are going to have a sticky end with it. So this is adapter. So this adapter already having a So it is already having a sticky end with it. This sticky end is there. So it means if such type of a creation is there in which the sticky end is there, if you synthesize a oligonucleotide which having a sticky end, to that you are calling it as an adapter. So this is going to have one blunt end and one sticky end. So you are joining this adapter to the DNA molecule that you are having. So this is your DNA molecule. This is double stranded. It is having this blunt ended one. So by this blunt, because of this blunt end, you can't ligate it efficiently. So what we are doing, we are adding this one. So this one means you are adding adapter to it. So you are adding an adapter to it. So this is going to be adapter. So this adapter is going to be having an overhang. So this is adapter. On the both hand you are going to be.
Okay, sir.
But in case of linker, what is going to be? So linker in case again you are going to have a DNA. Okay, this is a double stranded one. Okay, so you get double. So you are adding a linker to it. So linker is going to be again a double stranded one. Okay. So to this
Okay,
double
sir.
stranded one, you are going to add a restriction endonuclease RE. So when you add the RE, what is going to be? So in this, there is a restriction site is present, like here and there. So when you restrict it, it is going to produce overhanging one. But in this case, you need not to treat it with the restriction in there. This step is not required. Whereas this is must to generate a sticky end. Okay.
Okay, sir. Thank you.
Okay. So any other questions?
Sir, between
Sir,
adapters and linkers, which is more preferred?
Adapters are being more preferred.
Sir,
Sorry,
why is
linkers.
it?
Sorry, linkers.
But why, sir? Because I mean, with linkers, you have the extra restriction enzyme step also.
See, when you talk about the word which is going to be preferred, both are going to be preferred depending upon your requirement, what you need. But then even nowadays, we even don't need these linkers and adapters. We try to do blunt end cloning. Also, the thing is that that I mentioned terminal nucleotide transferase. We also try to use that also. So nowadays we are having multiple approaches. So now these linkers and adapters are a bit old approach. Okay, so depending on your requirement, we are using both. But as I mentioned, what I said, sorry, this adapt. Uh, you are using these linkers more as compared to the adapters because in the adapters there is a no as you've seen in this when we talk about the drawback. So such situation is going to occur a self annealing. So in this case, what is happening? In this case, we are going to lead with the again the blunt-ended one. So that is not the case with the your when you are using linkers. So in that case, you are using restriction enzyme. So once you done with it, but in that case, you need to take the precaution that the adapter that you are using, you must not use a site which is present in the linker, which is also present inside the your DNA fragment. Because if it is there, then when you are using restrict enzyme, it is going to break your DNA also.
Okay, sir. Thank you.
Sir, I had a question.
Yes, Yukta. Manashri, one more thing.
Uh,
Nowadays, we also use the TA cloning. So when we are going to talk about the cloning, so at that time I will also further explain your question. So which is going to be better and what different approaches we are using. Yes, Yukta.
Okay, sir. Thank you.
Sir, how having more recognition sites in the blunt and in molecule is a problem, is considered a problem? You mentioned when you were talking
Okay.
about linkers.
Okay. So okay. So this is a linker. So here this PMH1 site is there. You are incorporating this PMH1 site. So now what is there? So here is a you are joining this linker to a DNA that you want. So if in this DNA fragment, if you are having a BAM site also, means this GGA-TCC sequence is going to be there also. So when you restrict digest this linker with the BAMH1, so then what is going to be? It is going to put a cut here to generate the sticky end that you need, but it will also create break here. So it is
Yes,
doing
sir.
this one. So then in that case, your fragment of interest that you want to incorporate into the vector, that will be lost. Okay.
Yes, sir. Yes.
Yes. Next.
Sir, I have another doubt.
Yes.
Why are the linkers, why do they have to be added in high concentration?
See, as I mentioned in the start, we want to make your blunt end sticky because the efficiency of blunt end ligation is poor. Okay.
Okay,
So
sir. Okay.
So that's why if you use in a large amount, so around your DNA fragment, the availability of the segment that you want to ligate will be higher. So ligation efficiency will be better. So if you are having 1 to 1, then in that case, what is going to might possibility that your two DNA segment that you want to like this to which do you want to link the adapters? they may also join. Then what is going to happen? The fragment, like it is not going to be in this, like this is the one in which we are referring. So in that case, just a moment. So then this is a DNA fragment. This is a adapter you want to link. But what is going to occur? This fragment is going to link with itself also. Then the fragment size will increase. It will become double the size. Because your DNA ligase is going to treat blunt two DNA ends equally. It is not going to bias between the molecule to which you want to add linkers, or you want to join two linkers together. So what way you are joining? Ligase is going to not going to bias. So it may ligase this to this, or this to this, or this to this. But what we want to make that linker must ligate to your DNA molecules. Linker must ligate to your DNA molecules. So in that case, what we can do, we can't increase this concentration. So or what we have in the excess, we have your linkers in the excess. That's where we are using large amount, so that ligation mixture or ligated product will be of your desire.
Okay, sir.
Next. Okay, so if no question from linkers and adapters, now this whole module is open. So anything throughout this module that what we have done, if you would like to ask, please raise your questions. Keeping in mind, Monday is your test. So we can
Sir,
do one thing. So we can, yes, Yukta.
And sir, what are PCR inhibitors? What enzymes are those?
What are
PCR inhibitors.
PCR inhibitor will be talked when we were going to talk about the PCR. PCR is, we haven't talked about PCR in this module.
Sir, actually, it was I it was on LMS when I was studying module 1. That's why I asked. Okay, we'll discuss it later.
This PCR is not there in it. So PCR inhibitor, if you want to talk about, so that we can also talk about in it. As you mentioned that in case when we're talking about the DNA polymerases.
Yes.
Okay. So anything which is going to inhibit the activity of DNA polymerase, simply we don't call them the PCR inhibitor for this point of time. At this point of time, simply just say that it is a DNA polymerase inhibitor. So those who are inhibiting the activity of DNA polymerase, they are going to prevent DNA replication or DNA polymerization. PCR is also a polymerase, as we name the PCR is a polymerase chain reaction. So PCR is going to utilize thermostable DNA polymerases. So the thermostable DNA polymerases are there. If any substance, any molecules that are inhibiting the activity of thermostable DNA polymerase will be termed as a PCR inhibitors also. Okay. So that's why when we were talking about in your laboratory, when we mentioned that your DNA must be pure, because the DNA, if it is going to have some impurities, like we know that it is going to have, might be DNases are going to be there, or some molecules are also present in your cells. So cell is preventing the activity of polymerases, because it is allowing of action of the polymerase only when it have to go for the replication. So other than the replication process, cell do not want to go for replication. So but DNA polymerase might be there. So at that time they are producing some molecules which are inhibiting the activity of DNA polymerases. So those molecules which are present in your DNA sample, if you keep them, then they are going to inhibit the PCR also, to them we call the PCR inhibitors.
Okay, sir. Thank you.
Okay, so now I'm giving you a 5 minute. So just go through what we have talked about. You can visit the slides also, which I have shared to the LMS. Just go through them and also go through what we have talked about, or might be some of you are also making notes throughout the lecture. If you are not doing, then maybe it's your fault. Or in that case, you can go to the presentation that is there on the LMS. And after 5 minutes, I will again take your questions. If you found that there is no specific doubt, then we'll conclude our today's session. Okay, so I will come back after 5 minutes. So please go through whatever we have done yet, and I will take your questions. Okay, so now it is 5:52. We will reinstate. Be in the team, don't leave the team, and come up with the question at 2:57. Okay, students. So now I think you revisited some of your slides. Okay, so any query? Because from Monday, some of you are going to join the campus. So the classes are going to be in a hybrid mode. So those who are going to come here, they are going to be in front of us, and those who are going to be definitely are going to be offline. Online. Okay, so test will be in similar way. So those who are here, they will give an offline test, and those who are on computer, they will give online. So no query. किसी का कोई सवाल कोई जवाब नहीं? Manishwini,
No, sir, no queries.
everything is clear?
Yes, sir, it's clear.
Okay, so Yukta.
No, sir.
Okay, and Nehal.
Sir,
Yes.
sir, what are non-canonical restriction sites?
See, when we are saying the non-canonical means if we are going to use something like restriction nucleus 1 or 3, that type. And there also means as I mentioned, some table that you've seen will show you.
Sir, in star activity in your PPT, the non-canonical site is mentioned.
That's what I'll show you. So see, so these are the sites which are there, which are going to be recognized by these sequences. Like eCarbon is going to recognize GATTC. So this is its own site. But if it is recognizing other than, which is its site of recognition, that you will call it as a non-canonical one. It's recognized it, so it is recognized it, but under the different condition.
Okay, sir.
Okay. So this the difference between the canonical and non-canonical simply is what site it is recognized. It means traditional and non-traditional, you can say. Okay.
Okay, sir.
Any other thing?
No, sir.
Okay. So Shakti. Vivek. Vivek.
No, sir, no queries.
Janvi.
No, sir, no questions.
Okay, so if there is no question, then we can conclude. And before concluding, its attendance is must. So just wait for a moment. So when I call your name, please turn on your camera. Demetrius.
Present, sir.
Tridev.
Present, sir.
Manaswini.
Present, sir.
Adish.
Present, sir.
Yuvraj.
Present, sir.
Sanjana. 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 Nehal.
Present, sir.
Atoiba.
Present, sir.
Devanshi.
Present, sir.
Nandini.
Present, sir.
Kshitij.
Present, sir.
Janvi.
Present, sir.
Shivani. 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 Shakti.
Present, sir.
Rupal.
Present, sir.
Sahiba. Sahiba.
Present, sir.
Ishita.
Present, sir.
Namrita.
Present, sir.
Tanvi.
Present, sir.
Yukta.
Present, sir.
Ayushi.
Present, sir.
And Vivek.
Sir, I'm also present.
Present,
Shivani.
sir.
आप उस समय कहाँ थे भाई शिवानी? शिवानी तब कहाँ थे?
मेरा माइक अनम्यूट नहीं हो रहा था, हैंग हो गया था। आई एम सॉरी सर।
बट ऐसे ऐसे वो उसी समय कैसे हो जाता है ये?
सर मैं हर बार ऑन टाइम ही बोलती हूँ, एक्चुअली म्यूट अनम्यूट नहीं हो रहा था।
एंड संजना के साथ हमेशा ही होता है, संजना एंड युवराज. Sanjana and Yuvraj, this is the last warning for you people. If the mic stops working and camera also stops
Sir, I
working.
am present.
Yes, you
Am
are
I
present.
audible, sir?
Yes, you are audible. But why you are not responding earlier?
No, sir. I spoke, but you didn't heard.
And Sanjana, what's about with you? Sanjana? Sanjana? See, Sanjana is not responding even now. It means Sanjana should be considered as absent. Okay, so students, stay safe, stay happy. Good day and best wishes.
Thank you, sir.
So those who are coming to the
Thank you,
campus from
Thank you,
Monday, so welcome to all those who are coming. So see you soon.